Hi class, a couple quick things that I want to make clear about our recent acid and base lab that you might find useful. First is our definition of what a buffer is. A buffer is best defined as something that maintains a set pH in a solution. So for instance, in our stomachs, we've got a set pH of about 2. So if you eat something really acidic, buffers keep it near 2. If you eat something basic, it keeps things near 2. Most buffers are a weak acid-base pair that can reversibly either accept or donate hydrogen ions to a solution. A couple of you guys try to tell me that buffers neutralize solutions. That's not quite the best way to talk about a buffer, because a buffer is not always going to push things towards pH 7. Again, a buffer is going to maintain some set point. So each buffering solution may have a different set point. So that's something that we want to make sure that we're really clear about with buffers. Second, we want to make sure that we understand the difference between qualitative and quantitative data points. Qualitative means anything that is not numerical. Colors, feelings, shapes, arrangements, those are all qualitative. Quantitative, you can hear that word quantity in it, is anything with actual number. I'm 5'4", I have 3 kids, right? Those numbers, those are quantitative. I think that'll help us as well. Also, lastly, I wanted to mention is this graph. You guys did a great job of figuring out how to embed it and get it into your reports. With this buffering graph, I would be very comfortable with understanding how that graph was developed, how to read those kinds of graphs before we take the next practical exam. That's a great sort of I learned how to do something in lab moment. Lastly, I want to look ahead to this week where we're covering macromolecules. This is a lab that is completely dry. You don't have to do anything this week. A lot of students have troubles with the very first part of the experiment where we look at reducing sugars. And it asks you for any of the tests that are positive in reducing sugars to list which reducing sugar is actually in the substance. There's a list of 4 potential reducing sugars that are given for you on the worksheet. They are, let me make sure that I write it, give them to you the right way, glucose, fructose, lactose, and maltose. There is no way that you're supposed to be able to figure this out visually. It's not by looking at one orange versus a different red you can tell if it's glucose or maltose or lactose. Instead, I'm asking you to make some connections with what you already know, or you might have to look up some stuff. So when a substance tests positive for a reducing sugar, it's up to you to sort of logically think through and think, hey, does this have glucose in it? Or might this have lactose in it? Or might it have fructose in it? Or might it have maltose in it? I'll give you one helpful hint. When starch is broken down into disaccharides, one of the first disaccharides it's broken down to is maltose. Hopefully that'll help a little bit on this week's lab. Hopefully everything's going well for you in class, and I encourage you, if you haven't done so already, to reach out to your lab group where you'll be writing your formal lab report that is coming up due here in a couple weeks, and I want to make sure that you worked on that together with your lab members. Hope everything's going well for you, and definitely contact me if you've got any questions about things. Thanks. Bye.